that I could show you right now, which I don't have. I will post it later today. So as I said in the email last night, the exams are graded and available. I was rather disappointed in the average. To be honest, it's the second lowest average I've ever had. So I'm not quite sure. And it's easy for an instructor to blame students, and I think it's easier for a student to blame an instructor. And I think, no, seriously, and I think that when an exam performance is not good, I think that the blame does go both ways. And so I think it's important that we spend some time at some point thinking about how we might change that, how we might improve that, because I really don't want you to have averages of 53 on an exam. So looking through it last night, I was, as I said, kind of surprised and disappointed in some of the things. Some of you did well, some didn't. The range was from 11 to 98, okay? So a pretty incredible range. And my experience with exams is that when one is too high, the next one people get complacent and it's usually too low. And conversely, when things are too low, usually they turn back up, and I think people get more serious about that. So I hope that's what happens. I know I'm certainly committed to doing whatever we need to do to get that average up there. And I know I've been a bit busy in my schedule, and it's been hard to see me. But that won't be the case for the next exam. And if I can be of help to you, I would gladly be so. Or if I can help you to find a tutor or whatever, let me know. Because I really want to see this is important material for you as you go off to your various careers, medical school, dental school, pharmacy school, whatever school that you're ultimately going to go do. So if I can be of help, let me know. Please don't be a stranger with that, okay? I got everybody quiet. You guys, I always say you guys are the quietest class I've ever had. I've never had a class that just sat here so quiet. It's amazing. So, okay, so the exams are available for pickup in the Biochem office. That's ALS room 2011. That's not my office. So some of you have been looking for it in my office. It's not. It's in the main Biochem office, ALS 2011. So please pick up your exam. There is a key posted outside my office, and you can take a look at the answers that we had and so forth for that. If you have questions about your grade or concerns about your grade, of course, also please come see me. Please let me know. Okay? Okay. Let's dive into the material. And we're going to finish up today talking about mechanisms of catalytic action. And then we'll turn to the first of our series of talks about controlling enzymes. And just as I hope you've seen that understanding catalysis is interesting and useful, so too I hope you will see that the control of enzymes is also very interesting. Well, last time I got started talking about restriction endonucleases, and I'll remind you that restriction endonucleases we also call restriction enzymes. In fact, that's the most common name they have, is restriction enzyme. Researchers use restriction enzymes to cut DNA at specific sequences, and they are very specific for those sequences. When the restriction enzyme binds to a sequence that it recognizes, it will actually initiate cuts at that sequence, and there are two cuts, one on each strand, that result in the linearization or the cutting off of a fragment at that site. So that's a very useful thing if we're doing biotechnology in the laboratory. Okay? Now, each enzyme has its own specific sequence that it recognizes, and the sequences that it recognizes typically are between 4 and 8 base pairs long. These aren't things that you need to write down. I mean, I'm just telling you some facts about them. But the important thing is that they are specific sequences. Now, what I'm going to go through is tell you about one mechanism that one enzyme uses. And I picked this because, again, it's reiterating this theme that we've been seeing on the other enzymes that I've been talking about. And I think you'll see what I'm talking about briefly. We're not going to go into detail on this mechanism like we did with the chymotrypsin mechanism. Okay? So you can rest assured with that. Well, as I said last time, enzymes, the bacteria have the enzymes as a way of protecting themselves against invading viruses. So viruses that infect bacteria are called bacteriophages, and they work by attaching themselves to a bacterial cell and injecting their nucleic acid into the cell. That nucleic acid gets translated into protein, and that protein can replicate the virus. That protein can make a coat to make new viruses. And then after a period of time, that cell explodes, and all these viruses that were made in there are released. So bacterial cells have the same battle against viruses that we do. And as I noted last time, one difference is that because bacteria are unicellular, they do not have an immune system. So this is their sort of equivalent of an immune system. This is the main protection they have against invading viruses. So as I mentioned, what will happen is a virus, when it injects a bacteriophage, when it injects its DNA into the bacterial cell, is not methylated. It doesn't have a methyl group put on it. The bacterial cell has restriction endonucleases that recognize specific sequences. Those sequences are found in the viral DNA. And the viral DNA generally gets cut up. Okay? Well, when that happens, of course, the virus has been disabled. The virus can't function. And the bacterium is protected. Okay? Now, if by chance the methylase, which is the enzyme that is also made by the the bacterium, makes it to the phage DNA first, then it puts methyl groups on at those specific sequences and prevents the restriction enzyme from cutting. Okay? So, in rare cases, the virus actually escapes that kind of cutting. Yes, ma'am?
Wouldn't that be a problem in DNA replication as well of which one gets there first?
So her question is would that be a problem in DNA replication depending upon which one gets there first? It's not really a problem because the methyl groups do not interfere with the replication of the DNA, only the cutting of the DNA. Now, there are some slight problems that happen with respect to the methyl groups being on there that are chemical in nature, but it doesn't really have a significant impact on replication. But it's a very good question. Yes?
Like the bacteria, or one of the viruses that have the methylates on them replicate,
Yep.
they also have the methylates on them?
Good question. So once they replicate, do they get methylated also? What do you suppose? Yes. Yeah, once they've sort of gotten in the environments of the cell, they pretty much are protected. So just like those methylases are there protecting the cellular DNA, they would be protecting the viral DNA as well. And so the virus pretty much escapes that detection. We could also imagine that there might be some problems where, well, what if the restriction enzyme gets to the cellular DNA before the methylase gets there? What's going to happen? The cell's going to be hosed, right? Because it just chopped up its own DNA. So it's not a perfect system, but it's a pretty darn good system. It gives pretty good protection to the virus. Well, that wasn't what I started talking about this about. I wanted to give you this example and then tell you something about how it works. So let's talk about how a restriction endonuclease works. Most restriction endonucleases recognize what are called symmetric sequences. Now, what is a symmetric sequence? Well, look at this sequence recognized by the restriction endonuclease EcoR5. The top strand goes from 5' to 3', GAT-ATC. The bottom strand, look at this, it goes GAT-ATC. It's symmetric. It's what we call a palindromic sequence, even though it's not truly a palindrome. That's what molecular biologists call it. Okay? So most restriction enzymes recognize symmetric sequences, and that makes it nice because in this case, it cuts the enzyme cuts between the A and the T, not where that guy is, but between this A and this T. Well, that means it cuts between this A and this T on this strand, and the same A and T on this strand, and so it's one enzyme cuts both sequences, and both sequences are identical as far as the enzyme is concerned. Now, the asterisk that's on there is the place where the methyl group gets put on. And the placement of that methyl group onto that sequence keeps the restriction enzyme from recognizing that sequence. This restriction enzyme no longer recognizes that as a GAT-ATC anymore, and so it won't cut there. Well, what if the methyl group is not there, and what happens then? That's what we're going to talk about for this mechanism. Okay? Well, what's happening with a restriction endonuclease is that phosphodiester bonds have to be cleaved. We'll talk more about them next term when we talk about DNA, but the bonds between adjacent nucleotides and DNA are phosphodiester bonds. Okay? So the restriction enzyme is cutting those bonds. It's breaking those bonds between adjacent nucleotides. Okay? I'm going to show you how that works. And I think you can actually probably figure some of this out yourself, but I will step you through it. Okay? I'm not going to step you through that. In fact, I'm going to go through this relatively quickly. I'm not going to step you through that. And I'm not going to talk about that. What I want to talk about briefly is magnesium. Okay? So virtually all restriction endonucleases require magnesium to function. Most DNA, in fact, has a coating of magnesium around it. The reason for this is DNA is very negatively charged and magnesium is very positively charged. So it's hard to find DNA without magnesium around it, which works very nice for the restriction endonucleases. Okay? This is simply showing you again the symmetry that I showed before. There's the GAT-ATC, GAT-ATC, and the green dot there is where the cleavage actually will occur. If we look at the sequence of the two strands, we see that this cleavage is occurring like right, let's see, we've got GAT right here. This X marks the place that each one of those strands will get cut. Okay, now, unfortunately your book does not have any really good figures, so I have to sort of step you through this. I know I'm flipping through these, I apologize for that. This is the best figure that your book makes for how this enzyme works. All right, so you're not going to draw a structure, at least nothing complicated for a structure for this enzyme. Let's imagine I'm a restriction enzyme and I recognize a GAT-ATC. How do I find my GAT-ATC so that I can cut it? Well, it appears the restriction endonucleases work as follows. They have an affinity, an ability to bind DNA that's of any sequence. So once they see a double strand of DNA, they grab a hold of the double strand. Okay? So they've grabbed a hold of the double strand, and they look. Have I just bound a GAT-ATC? Well, most likely, by chance, they have not. GAT-ATC occurs by random possibility once every 4,096 nucleotides. Okay? So if it's bounded and it hasn't found GAT-ATC, it starts sliding along the double helix. And as it's sliding along the double helix, it's matching up components of itself with the hydrogen bonds on the individual base pairs. Okay? So it can actually read the sequence as it's going along by telling what hydrogen bonds it's forming. It goes sliding along, sliding along, sliding along, and all of a sudden it hits, in this case, a GAT-ATC. Okay? It's been reading the sequence. Now it recognizes, okay, I have a GAT-ATC in my active site. When we had chymotrypsin was moving along until it got a phenylalanine in it, right? In the S1 pocket. What happened when chymotrypsin got the phenylalanine in the S1 pocket? Made the alkoxide ion, but why did we make the alkoxide ion? We had a shape change, okay? Restriction endonuclease is exactly the same way. We've got the right substrate, and the restriction endonuclease undergoes a shape change. And guess what? That shape change bends the DNA. That's what they're trying to show you right here. The DNA is being bent within this structure here. Okay? So the bending only happens when the restriction enzyme recognizes the proper sequence. All right? Now, why is that important? Well, it turns out that the bending causes a magnesium ion to be positioned in exactly the right place under the DNA so that it can take off a proton off of water. Okay? The magnesium ion facilitates the removal of a proton off of water. It's not a charge phenomenon, but it is removing a proton, and guess what that proton leaves? A nucleophile. And guess what that nucleophile does? It attacks the phosphodiester bond. That's how you break a phosphodiester bond. Okay? Now, I'm simplifying several things. If you look in the book, you're going to see there's a more complicated mechanism than what I've just told you. So I'm trying to give you a mechanism that parallels what we've talked about before and keep it in simple terms. And I'm only going to hold you responsible for those simple terms. The steps in the process: binding to DNA, sliding and reading the sequence, finding the right sequence. When that happens, there's a bend. The bend now puts together a perfect architecture so that that magnesium ion can now help in the activation of water, which creates a nucleophile, which attacks the phosphodiester bond, and bang, we've broken the DNA. When the wrong sequence is there, that doesn't happen, and that's why restriction enzymes only cut when they hit a specific sequence. Yes, sir? Is that why we use EDTA in so many buffers? You use EDTA to help control magnesium concentration. The answer to that question is partly yes. Yeah. Yes? Well, that will have to get more into mechanism. If you'd like, I can sit down and show you that. But there's a more elaborate mechanism than what I'm there. But the magnesium plays a role in the activation of that water by helping to get that hydrogen pulled off. Yes? Does one enzyme cut both phosphodiester bonds? The answer is usually yes. And most enzymes... I believe you said Happy Halloween. But personally, I would be reluctant to eat that candy, folks. Okay. Everybody okay? I came, I saw, I treated, right? There's a new one. You guys haven't had a story. I'll tell you a story. Was anybody here last year? Was anybody in this class? Well, you don't have to raise your hand if you were, but last year, it's a true story. So I'm up here lecturing, and there's these two guys who think they're really smart, okay? And they're dressed as two old men, and they come sauntering in here, up to the front, and everybody in the class is watching them, okay? And so they're so tickled because they've gotten all this attention and everything, and they come up and they're doing this and so forth. And I'm a little unhappy because it's disturbing the class, right? But I don't say anything, and they sit down here, and so ruff, like this, and so forth. And I said, we have some visitors to our class today. And I said, we have two, gentlemen over here, and thanks to these two gentlemen being here today, you're going to have a pop quiz. Oh boy, did the attitude towards these two gentlemen change very quickly. and they start looking around like this, like, what's going on? And I said, these guys are the people that you have to thank for the fact that you have a pop quiz. Take out a piece of paper. Okay. Well, everybody started rustling, and a few of them made comments about these guys. Well, they quickly get up and go out of here. Okay. And so I said, okay, on your piece of paper, write your name, and we'll have extra credit. Well, of course, they didn't realize that I wasn't going to give a pop quiz, but they thought they were to blame for the reason they had a pop quiz. And I thought, hopefully they learned something about that. You should not really disturb classes like that. It's not a good idea. I'm the only one that should disturb classes like that. So anyway, that was cute. Okay. So everybody got the restriction enzyme, how it works? This is the most relaxed I've seen the class in a long time. Maybe I should have them come back. Okay. All right. So that's what's up with a restriction endonuclease. Yes?
On the last picture was the pink line, the DNA, and the big structure around the enzyme.
Okay. So what you're seeing here is actually the DNA, and then this is the sort of central helix of the DNA in red. Okay, so you're seeing bases that are here, and you're seeing a bending that's going on. They're not depicting the protein here so that it's easier to look at. Now, well, why does methylation stop this process from happening? Two things. One, the enzyme doesn't fully recognize it as the proper site, so it doesn't bind properly. And second, that methyl group keeps the magnesium from getting positioned properly. Therefore, even if it does stop there, the enzyme can't cut it. So that methyl group plays that critical role in protecting the enzyme from being able to cut. Yes, ma'am?
So does it only require one methyl group to stop it?
Oh, good question. Does it require only one methyl group? Because remember, you've got two strands there that have the same sequence. Does it require only one methyl group to stop it? It depends a lot on the enzyme, but most enzymes will cut the strand that's not methylated if only one is. So that's actually used as a technique in molecular biology to do what's called nicking DNA at specific places. So the answer is, but it does depend on the enzyme. So not all enzymes are identical in that respect. Okay. The last thing I'm going to say about enzyme mechanism is very basic, very simple, and we're not even really going to have mechanism per se. This, and the reason I bring it up is we'll talk about this group of enzymes a little bit later. They're called nucleoside monophosphate kinases. Okay? You can also abbreviate those as NMP kinases, nucleoside monophosphate kinases. These enzymes play a critical role in the synthesis of nucleotides. They convert nucleoside monophosphates to nucleoside diphosphates. That's a mouthful way of saying they can convert, for example, AMP into ADP. In nucleoside, they play a critical role in nucleotide biosynthesis. Yeah. So they will convert something that has a monophosphate to a diphosphate. AMP goes to ADP. Other enzymes will convert ADP to ATP, and now ATP we can use for energy. Okay? Now, these enzymes, the only reason I mention them is because they have an interesting mechanism that involves, okay, something called a lid. Okay? Here we go. Now, here's the enzyme structure, and you can see the enzyme structure in the absence of ATP. ATP is used to donate the phosphate. So ATP, the phosphate that goes onto AMP, comes from ATP. All right? So I need AMP plus ATP goes to 2 ADPs. So a phosphate is moving from ATP onto AMP. Okay? Everybody got that? All right. So the reaction they catalyze, an example, there's many different reactions, but an example reaction could be, let's say instead of using AMP, let's say I've got CMP, cytosine monophosphate. CMP plus ATP, the phosphate moves from ATP onto CMP, so I get CDP plus ADP. Okay? So I'm just transferring a phosphate from one side to the other. All right? Now, we've heard, and you all have heard from me telling you on and on, about the fact that binding of substrate causes shape changes in enzymes. Look at this shape change. This guy already has bound to it, it's not shown, but already has bound to it the monophosphate. So in this case, you've got CMP bound. When ATP binds, look what happens. This guy has what we call a lid that actually closes on the active site. The closing of the lid is important in this catalytic mechanism, as I'll tell you in a moment. Okay? So the binding of ATP is causing not even just a slight shape change, but a pretty good size shape change, and that shape change is closing the lid. Well, for example, why do you close the lid on something that you have a box of? What's that? To contain it. You don't want things coming out, right? You know, guys don't close the lid on the toilet, right? Well, that's a different thing, I guess, right? But, all right, but the point is, that was getting disgusting. I didn't mean that to be quite that way. I thought, hey, there's a lid, right? You know? Okay. The point is you close the lid on something to contain. Maybe the toilet's going to overflow. I don't know. But I guess the lid doesn't stop that, though, does it? So, okay. Get off of that. All right. We close the lid to keep things in. That's why the enzyme closes its lid. All right? Well, what happens is when the phosphate is moving from ATP over to the monophosphate, in this case CMP, that phosphate can literally escape. It can get away. With the lid there, it's got no place to go. And that's why it goes from the triphosphate directly over to the monophosphate. The enzyme is not set up to contain it in any other way except for using this lid. And the lid is very, very effective. Okay? Makes sense? Yes, ma'am.
So it only takes the extra phosphate from ATP, or could it also take it from something else?
So our question is, can the phosphate only come from ATP, or could it come from GTP or CTP? Generally, it comes from ATP, since ATP is the most abundant one. There are some that may use other triphosphates, but that's not really essential for our knowledge here. Okay? Okay. Well, that's the last of what I want to say about mechanism. Any other questions before I move to the next material? Okay. let's turn our attention then to talking about control of enzyme action. So, I've said several times, and I'll repeat it here, that enzymes are powerful things for cells. And power needs to be controlled, because just like the Maserati that you take to the grocery store would cause a problem if you ran it too fast and everybody had one, so too do enzymes have to be controlled, because if we don't control those enzymes, they're going to use up the cell's resources too fast, they're going to make too much of something that we might need a little bit of, etc. So being able to control enzymes is very, very important. We're going to talk this term about 3 different ways of controlling enzymes. Okay? You're seeing the first of them right here. It's called allosterism. Now, I gave you a definition earlier, but I'll repeat it here. Allosterism occurs when a small molecule binds to a protein and affects the protein's activity. A small molecule binds to a protein, I say a protein, but binds to an enzyme. A small molecule binds to an enzyme and affects the enzyme's activity. Okay? Yes?
So, would say that hemoglobin doesn't exhibit allosteric activity?
Yeah, that's why I actually changed it to say enzyme, because hemoglobin does not exhibit allosterism, it exhibits cooperativity. Yeah.
The book
Isn't
says
it?
exhibits allosteric.
What's that?
The book says it exhibits allosteric.
Yeah, the book I don't agree with, so, and most people don't, because it requires an enzyme for it to be allosterism. You don't want to be able to, well, it's a semantic argument, but the fact of the matter is that enzymes have to be controlled, and that's a mechanism. Okay. Okay. Now, that's one thing to consider. A second mechanism for controlling enzymes is covalent modification. Covalent modification. That might involve breaking of a peptide bond in some cases. That might involve addition or removal of phosphates. We'll see a lot of examples of that. Or in some cases, other rarer things. Okay? So, allosterism 1, covalent modification number 2. The third mechanism, you're going to go, well, duh, all right? The third mechanism is whether or not the enzyme is made. If the enzyme is not made, then of course you've got perfect control. Now that becomes important when we consider some processes like gluconeogenesis. Gluconeogenesis is the way that our body makes glucose. Not all cells of our body make glucose. Our liver and kidney are the primary places our body makes glucose. Why? Because a couple of the enzymes that are critical for the process aren't made in all cells. They're only made in the liver and in the kidney. Okay? So, control of enzymes can also occur at the level of whether or not, or control of enzymes, in this case pathways, whether or not the enzyme is even made. Okay, well let's focus our attention first on allosterism. Allosterism is the binding of a small molecule to an enzyme to affect the enzyme's activity. The classic molecule that we describe, the classic enzyme that we describe this with is known as ATCase. You're going to hear a lot about this enzyme both this term and next term. ATCase has a longer name. I'll give it to you, but you're not responsible for it. It's called Aspartate transcarbamylase. Big mouthful of a name. The spelling's in the book if you want it. Make sure if you use the abbreviation ATCase, you call it ATCase. We do not accept ACTase, for example, which is a common misspelling of the abbreviation. Okay, ATCase. ATCase catalyzes a reaction that you see right here. Okay, you notice I didn't even give you the molecules. One of the molecules up here is aspartic acid. That's the aspartate part of the enzyme name. Okay, the names of the molecules aren't even that important for us. All right. This catalyzes a reaction. This reaction is important because the product of this reaction is used to make another molecule. And that other molecule is used to make a third molecule. And the third molecule is used to make a fourth molecule. Okay? There's a series of 10 such reactions. And at the end of those 10 reactions, we end up with the nucleotide CTP. What I've just described to you is what we call a metabolic pathway. A pathway occurs One reaction leads to a second reaction, leads to a third reaction, leads to a fourth reaction, et cetera, et cetera. Okay? There's a link. The product of one reaction is the substrate for the next reaction, which makes a product that is the substrate for the next reaction, which makes a product that is the substrate for the next reaction. Okay? Well, why do I tell you about this? All we're interested in is ATCase. Well, this enzyme catalyzes the first reaction on the way to making CTP. The first reaction on the way to making CTP. The end product, once this guy gets made, is that there's 9 more steps and we get down, we make CTP. Well, we'll talk a lot next term about the fact cells are paranoid about making too much of a given nucleotide. If they make too much of a given nucleotide, they are way more prone to having mutations. Way more prone. So cells are paranoid. They have more controls about levels of nucleotides than they do on any other metabolic pathway. They're very careful about not making too much of any given nucleotide or too little of any given nucleotide, because if they do, there's much more likely they're going to mutate, and mutate can lead to cancer and other problems for a multicellular organism. Okay? What I'm getting ready to describe to you is one such control. All right? So we take as principle that cells don't want to make too much of a nucleotide. That means that ATCase is one of the enzymes that needs to be controlled. Now, what we discover is when we have a metabolic pathway, cells do not typically control every enzyme in the pathway. That's inefficient. If they stop the very first reaction, then they stop the whole pathway. Because if there's no product 2, there can't be any reaction number 2 to make number 3. If we stop it right at the source, then we stop the whole pathway from occurring. And that's exactly what cells do for most metabolic pathways. We'll see some exceptions, but most metabolic pathways are controlled in just that way. The first enzyme gets controlled. Well, in our scheme of things, we start thinking about, well, how would the cell know when to turn off ATCase, and how would it know when to turn on ATCase? Well, you see part of the answer on the screen. When CTP concentrations in the cell start getting high, CTP binds to ATCase and allosterically inhibits it. It allosterically inhibits it. Allosteric inhibition is not typically on or off, although you'll probably think about that that way, like I do as well, but more like turning down the volume. That is, slowing the reaction, okay, instead of stopping it completely. But slows it quite a bit. This is a negative regulatory mechanism. That's correct. Okay? This type of inhibition has a name. It's called feedback inhibition. And it's called feedback because the last product is feeding back to the first reaction and saying, hey, hello, there's too much of me. Shut off my synthesis. So when CTP starts accumulating, this guy binds to the enzyme, and the enzyme goes, oh, I've already made too much. Slow down. Don't make any more. You've overfilled the gas tank already, and it's starting to spill out onto the pavement. This could be dangerous, right? Of course, we don't have self-serve in Oregon. Nobody knows that anymore, right? So, any of you who have ever had to fill your own tank might know that. You don't want to overfill the tank. Now, it's useful, yes?
So it slows the reaction down, but it doesn't fully stop it?
It slows the reaction down. Allosteric mechanisms typically do not completely stop it. Okay? But the slowing is very significant. Okay? Now, well, I said it's nice to be able to turn it off. Wouldn't it be nice to be able to turn it on? And in fact, this enzyme can both be turned off and turned on. And again, it's not fully on. It's just turning the volume way up. It makes it go a lot faster. If you're starting to think, does this involve T state and R state? The answer is, yes, it does. What state do you suppose CTP causes the enzyme to be in? T state. Okay? CTP converts the enzyme into the T state. Well, I'm a cell. I am looking for a little fun this weekend. Okay? So I've decided I'd like to replicate. Okay? I'm going to go do my thing, right? Well, how do I know if I can score this weekend, right? Or score tonight, right? You guys have dirty minds. Okay? How do I know that I'm going to be able to score tonight, right? Well, a very good thing will be it takes a lot of energy for this sort of stuff, right? I mean, pulling cells apart doesn't just happen. It takes some energy. You've got to replicate your DNA. That's going to take some nucleotides. It's going to take a bunch of stuff. Maybe I should use my energy level as a way of controlling whether or not I can replicate. Guess what? ATP turns this enzyme on. ATP says, we're going to score tonight, guys. We got to be ready. Okay? So when ATP is in great abundance, this enzyme gets turned on because... Turned on, get it? Okay. This enzyme... That was a bad joke. Okay. All right. This enzyme gets activated by ATP. Lots of energy. The cell's going to do something with that energy. Hey, I'm going to divide, and I'm going to need nucleotides to do that. So the decision that this enzyme has then is, do I have a lot of energy, or do I have too much CTP? And it's constantly balancing those two, going between the on and the off. Okay? As a consequence of that, the cell is able to control its synthesis of nucleotides, and as a consequence, control whether or not and when it divides. Yes, back there.
Is it a different allosteric site that binds CTP?
Good question. Is it a different allosteric site that binds CTP versus ATP? The answer is no. So I'll show you a structure of the enzyme in a second. The enzyme has 12 subunits. 6 of them are regulatory, which is what binds the ATP or the CTP, and 6 of them that bind the substrate, one of which is aspartate. Do you have a question?
So does the ATP kick off the CTP?
Does ATP kick off? Does ATP kick off the CTP? No. All of these bindings are reversible. So remember, once CTP is on there, it's not going to stay on there forever. It's going to come on, go off, right? So if there's a lot of ATP, ATP squeezes in, and there it is, right? Yes, sir. I'm sorry, I didn't hear the first part of what you said. Is the enzyme ever in a state where it has neither of those on? The enzyme, yes, can be in a state where it has neither of those on. That's correct. Yes, ma'am. The R state, Okay, so we see nice on-off switches with this enzyme. CTP turns it off by feedback inhibition. ATP turns it on. There's not a name for ATP turning it on other than just simply activation. Here's the reaction, in case you want names. Blah, 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 blah. Notice 3 arrows, actually a total of about 10 arrows that lead down to this guy down here. Aspartate is a substrate. I talked about aspartate being, what was I going to say? A substrate. Giving the enzyme its name, I guess that's what I said. So there's other reasons to mention this right here. Aspartate binds to the catalytic site of the enzyme. So I said we've got regulatory sites and we've got catalytic sites. Or regulatory subunits and catalytic subunits. Sometimes my mouth goes slower than my head does. Okay? If we look at this, we discover well, I'm behind. Okay. Here's what the structure I wanted to show you. Okay. 6 catalytics, 6 regulatory subunits. All right. The catalytics are shown here. They are a set of 2 trimers. We can think of underneath this trimer, there's 3 more just like those. So 6 catalytic, and you can see 1, 2, 3, 4, 5, 6 regulatory subunits. And now you're seeing it from the side, so you can see these individual ones. The regulatory subunits bind to the ATP or the CTP, and they change it from either the R state, as it's shown above, or the T state, as it's shown below. The T state, as you can see, is more compressed, and it's harder for the substrate to get into the T site, into the T state, which is why the enzyme is less active when it's in the T state. The substrate has a harder time getting to the active site. Okay. Now, what I was starting to tell you, let me show you some kinetics, and then I'll get to where I was going to start. That's why I got screwed up there. All right. Here is a plot of the enzyme velocity as a function of CTP concentration. Not surprisingly, as I add more CTP, the enzyme velocity slows down. Remember I said it doesn't turn it off, it slows it down. This is a log scale, so it's actually slowing it down quite a bit. Okay? The enzyme is slowing down as we add the inhibiting compound CTP. Here's something that will surprise you. There is something else that regulates this enzyme, and it's the substrate aspartate. Now, looking at this, what would this tell you? Okay, there's a clue that aspartate is not just going up, okay, as we see here, but it has a certain shape here, right? What's the shape that tells us something interesting is going on? The sigmoidal shape, right? And the sigmoidal shape tells us there's allosterism going on, but I haven't added any ATP or any CTP. This was exactly like what we saw when we had hemoglobin and we started adding oxygen. What happened? We saw that sigmoidal shape. This tells us that aspartate is acting like an allosteric effector as well. It's also converting the enzyme into the R state. So here's a substrate that's actually changing the enzyme, activating the enzyme, and causing it to go into the R state. Three things, therefore, regulate ATCase. ATP activates it, aspartate activates it, CTP inactivates it. Remember that aspartate is binding at the catalytic site. It's the only one that binds at the catalytic site. The other two are binding at the regulatory sites. Okay. Questions about that? Yeah, what I just said real quick again? Yeah. So, three things that regulate aspartate transcarbamylase, ATCase. ATP and aspartate both activate the enzyme. They both favor the R state. ATP binds at the regulatory subunits, and aspartate binds at the catalytic subunit. It's the only one that binds at the catalytic subunit. CTP inhibits, that is, favors the T state by binding to the regulatory subunit, just like ATP does, except for it's converting it to the T state, not the R state. Why do you suppose having aspartate activate the enzyme might be important? I made a joke about ATP. Can I make a joke about aspartate? I have a vote for a joke. Now if I can get somebody to tell the joke. Yeah, as a matter of fact, it makes very good sense because, again, abundance of amino acids tells the cell that, hey, we've got the materials here to help go ahead and do replication. So a lot of aspartate is a signal in the cell that, hey, we've got the materials that we need to go ahead. Let's get nucleotides made so we can go ahead and divide. So these little, very subtle signals that cells have, the enzymes have responses built into them such that they act accordingly. Okay. I have other things to say, but since it is Friday, I thought we would sing a song. Okay? Now, this song, I have to tell you, was inspired by an experience. This was inspired by an experience of a student who took an exam and didn't do very well on her exam. I'm not making this up. And so I wrote this for a student of mine, and so I want you guys to sing it with me. Anybody know the song, The Yellow Rose of Texas? Yellow Rose of Texas, da-da-da-da-da-da-da-da-da. Okay. This is called The Mellow Woes of Testing. Okay. It was written at the end of the term. The term is almost at an end, 10 weeks since it began. I worried how my grade was. I did not have a plan. The first exam went not so well. I got a 63. 'Twas just about the average score in biochemistry. 53 actually. I buckled down the second time, did not sow my wild oats. I downloaded the videos and took a ton of notes. I learned about free energy and delta G naught prime. My score increased by 7 points, a C+ grade was mine. I sang the songs I memorized, I played the MP3s, I learned the citrate cycle and I counted ATPs. I had electron transport town in all of complex V, and I guess when I saw my exam it was a 93. So heading to the final stretch, I crammed my memory and came to class on sunny days for quizzing comedy. I packed this card with info and my brain almost burned out. 'Twas much to my delight I got the A I dreamed about. So here's the moral of this song, it doesn't pay to stew. If scores are not quite what you want and you don't have a clue, the answers get into your head when you know what to do. Watch videos, read highlights, and review. All right. Okay. So, bottom line, don't get discouraged. Dig in, okay? How you doing?
This is kind of similar to that, but I didn't do very well on this exam. Is there still a chance for me to if I buckle down and
Well,
to
buckle
pull
down,
out?
buckle down. So it all varies on how you do compared to the average of what everybody else does. That's what.
Okay.
So is it possible? Sure, but you got to do it.
Okay.
Okay.
Excuse me, sir. It's I think I deserve the whole.
Okay. So you need to write. What does the syllabus say about requests?
I write a request.
Okay. So so give me the request then. I'll just take that. Okay.
Just the request or the.
Well,
the
without the exam, I don't have a way of looking at it, right?
Thank you,
Okay.
sir.
Hi.
Yeah, I know I have to put in a written request. I have a question about how you're not going to take points off if you think it's frivolous, because this one, I feel I should have gotten at least a point or two.
Well-
even if I didn't say KM.
Well, I can't tell you. I mean, if things are frivolous, I will take points off. That's what I will say. So hopefully it's not a frivolous request.
But what do you define as frivolous? Because it's not like I'm saying I just want
Well,
you to
how would I
read, want more
how would I
points.
define frivolous? I don't know how we would define frivolous, right? So the point is, if you think it's been taken off unfairly, then you should explain why. Okay, that's my guiding line.
I also want to know how you grade the grade for graduate student. Is it?
Okay. So the grades are set by the undergrads. So then your graduate student grades are laid on top of those.
So
they
we
set-
don't have a curve or
You will have the undergrad curve. The undergrads will set the curve.